25th webinar of our FCTL webinar series. We have our alumni who is currently a FulBright scholar in USA. So we all welcome Dr. Dibyakanta Seth, and today his topic is high pressure homogenization and approach to clean label food production. But before moving to everyone, happy Durga Puja and a very happy Navratri to all people in India, and obviously our speaker who is far away, but hopefully he have a festive, joyous weekend ahead. And before moving to anyone, obviously it's a tradition. We always tell our coordinator to tell a few words about his scholar with whom we have worked extensively. That's why I would request our Professor Mishra sir, to tell her his experience, his interaction with Dibyakanta sir. He's as far as I know, he's one of the most active, one of the most attentive scholars, and obviously a professor whom I know from Tezpur to NIT, and now he is a FulBright scholar. Professor Mishra, can you just tell a few words and we'll proceed our webinar?
Okay, thank you, Jayashree. Dear all, best wishes for the festival seasons. Happy Navratri and coming Durga Puja and Dasahara to you all. So Dibyakanta, as Jayashree was telling, he has always been a very sincere and hardworking scholar. He did, of course, MTech with me, but during the MTech, there was so much interaction and bonding that after MTech, I think he joined somewhere in the Northeast. And from there, he moved to Tezpur. And from Tezpur also, he continued our association, and in fact, he insisted he wanted to have me in his PhD team as well, and then he requested his PhD supervisor at Tezpur University, Professor Dept, to include me. In fact, I also I feel lucky to have been associated with him in his PhD work, which he did on spray drying of Mishti Dahi. Of course, before that, in fact, he had submitted one project to Ministry of Food Processing Industry, and which was finally obtained as the Ministry of Food Processing Industry. And even today also, now also, I keep on getting one or the other request from the journals that the paper which he published on Mishti Dahi powder and its drying, spray drying, etc., he has that paper has invited a lot of people, and a lot of people have become, and many journals, they want further research or presentations on those papers, etc. So after that, now he from Tezpur, he moved to NIT Rourkela, and presently, as Jayashree told, Dibyakanta is in US and FulBright fellow. So let's listen to Dibyakanta. That the topic today is very important, and I wish every success in Dibyakanta's life ahead. Thank you. Over to you, Jayashree.
Thank you, sir. So before going to any further delay, I would like to introduce our today our speaker, Dr. Dibyakanta Seth, who is FulBright Post-Doctoral Fellow in Department of Food Science at Ohio State University, and he is also currently serving as Assistant Professor in Department of Food Process Engineering, NIT Rourkela. Before that, he finished his BTech from OUAT in 2002, and then he worked as a research associate in dairy technology in NDRI. After that, he joined as an assistant professor in teaching and research in Kharagpur University, and later his stint was continuing in Tezpur University. There he worked for 10 years, and in between that, he gained his PhD from Dr. Dept and our coordinator. Later he joined NIT Rourkela, and he has been bestowed with Young Scientist, Young Educator Award in international conferences. He has been active member of AFST, Mysore, ICA, and finally Dr. Seth is now working as a FulBright scholar. So today I am presenting Dr. Dibyakanta Seth, and today his topic is high pressure homogenization and approach to clean label food production. Dr. Seth, over to you.
So thank you, Jayasri and Mishra sir, for the elaborative introduction about me. So thank you very much, and wish you all a happy Durga Puja. Today I will be going to...
Anyone who has a question can write their questions in the chat box. After the lecture is finished, we will again catch up here. Please mute your video and audio during the lecture.
So, is it visible?
No, your slide is not visible. You are not presenting. You go to the presentation mode. Keep full screen. Whole screen you share. Yes. Now it's fine. Yes.
Yes, sir. It's visible.
Okay.
That's fine. Yes.
Yes, it's visible.
So, thank you once again for the introduction. Today I'll be discussing about high pressure homogenization and approach to clean level food production. So, quite a few years now, almost a century years now, the term high pressure has come into our food research area. So, a lot of advancement has been taking place. So, I'll be discussing one of the advanced technology on that high pressure area and how it is helpful in giving us an opportunity to produce a clean level food. So, clean level food is now been a taboo. So, everybody are rushing into the clean level. So, this will be my outline of my presentation, clean level foods. So, we'll define what is clean level food, principle of high pressure homogenization. Then we'll discuss about some design considerations. So, what are the process conditions we can manipulate? What are the process conditions we can work upon to give different or to get the different variations of the food qualities. Also, we'll be discussing about the high pressure homogenization induced changes in the food products, and I'll be not going in detail of what are the changes happening in the chemistry level, chemical level, but it's overview I'll give, then future suggestions and conclusion. So the purpose of this today's presentation is just to acknowledge or just to let you people understand what is high pressure homogenization, what's the principle and their uses, and what are the things you can do or we can do in our future research. The consumer's need as far as the food is concerned is mostly based on the two things. One is self-life. The self-life of the food products should be high, first thing, and the food safety. The food safety is more concerned to the manufacturers rather than the consumers. Because they have to comply with different regulations. So for that, they have to be, the food which they sell has to be stable or safe. Safe in the sense, there should not be any harmful component or harmful organisms which will affect our body. And at the same time, we should have also the food products which should be tasty and nutritious. And nowadays, there is a paradigm shift of the consumer's choice about the nutritious and healthy foods. Earlier, consumers were not so aware of the nutritious or healthier foods, but because a lot of problems are coming, like so many diseases are actually coming, the health microbiota is getting deteriorated, so that's why people are getting aware of that, and they choose their food which are not only tasty, rather they should be nutritious and healthy. So this story doesn't end here. Even another one term has come, that is clean level. So that I'll discuss in my next slide. So what is clean level? So to accomplish all these things like self-life, food safety, and tasty and nutritious foods, we generally apply thermal processes. So in the thermal processes, of course, we can achieve the self-life, we can achieve the food safety, but somehow we have to compromise with the taste and the nutrition, because more the severe the heat treatment we give, the loss of nutrition will be nutritional components will be there. So we have to somehow optimize the processes. That's what we conventionally we do. So like pasteurization, sterilization, these are some of the processes. Sterilization is the harsh process where we can also achieve the killing of the spore forming bacteria. But at the same time, so we have to lose some of the valuable components. So how to maintain this cell life, at the same time, how to retain our most of the nutrition in the food components so that it will taste like the natural product or natural food material. So a lot of research has been going on, and the latter part, one term called that is non-thermal processing. So non-thermal processing, so without even using the thermal process like heat, conventional heat, so people use different other techniques so they can achieve all these goals like self-life, food safety, and tasty and nutrition. And it is so many research has been done, and it has been proved, validated that non-thermal processed foods are better in terms of the taste and the nutrition, along with the maintaining the cell life and the food safety of the product. So traditional non-thermal processed foods, the technologies are like the cold plasma, UV, pulse electric field, high pressure processing, and so many other things are there. So it is going to be a new generation technology, the intervention in the food processing now. Of course, the cost of production of the non-thermally processed foods are much higher today, but the manufacturers or the scientists are in a hope that yes, in coming future, so since when the mass production will be there, then the cost of production will go down, and it will be a part of their daily life to consume the non-thermally processed foods. So many non-thermally processed foods are also now available, like high pressure processed foods are available, because the scaling of the instruments are possible, or somehow it has been possible to some extent, and because of the scaling of the instruments, so people can produce or the manufacturers can adopt that technology, and they could produce the non-thermally processed foods. Like high pressure processing, also some companies actually they are producing the high pressure processed juices, so they can handle around 300 to 400 liter of milk per or liter of liquid per hour. So what is clean level food? So there was a survey asking what is clean level food. So 47% of the people they say they have never heard of. So of course I also didn't heard of clean level food before some time. 32% they say that yes, kind of heard of, but we don't understand what it is exactly. And heard and considered. And 17% they heard, then they have the little knowledge, and they are being influenced by the clean level foods. And assuming that this 17% will go to will move to 50% or 60% in the coming years, because nowadays in the globalist era, and also because of the strength of the social media, people are getting aware of all the things which they come across. And 4% of them, they are heard of, but they have no impact. Sorry. That means they heard of the clean level food, but they don't care about that, whether it is clean level or if it is tasty, then it's okay for them. So clean level food is actually not a scientific term, rather it's a consumer term. So it actually gives a trust to the consumers about the manufacturers. Manufacturers trust to the consumers, right? So whatever the manufacturer is manufacturing, so whether the manufacturer is giving the clear information about the ingredients that they are using or not, right? So the clean level food, as per definition, it refers to the products containing natural, familiar, simple ingredients that are easy to recognize, understand, and pronounce, with no artificial ingredients and synthetic chemicals. So there are a lot of discussion also. So everybody should not be familiar with all the components. Like we are very aware of vitamin E or vitamin C, but in the label, if it is written ascorbic acid, many of us also don't recognize, or the common man, they don't recognize what is ascorbic acid. If the labeling will be vitamin E or C, then they say that yes, we understand this vitamin C. So it is not necessary that all the ingredients manufacturers are adding. So all the people should be aware of that. But yes, being the manufacturer or being this the scientific community, our goal is to not to use any artificial ingredients and synthetic chemicals on it. So why we're worrying about that? Because all the synthetic chemicals and the artificial ingredients which you use may not give us a ill effect in the short term, but in the long run, so that actually gives many problems to our body health and so on. So that's why. So nowadays the scientific community, they are trying not to use the synthetic chemicals and ingredients, but that is not 100% possible, but they are trying their best to minimize at least the use of the synthetic chemicals. So clean level food is a food prepared in such a way that there is no chemicals, no artificial ingredients, and the ingredients they use, it's familiar to the customers or the consumers. So the global clean level ingredients market is expected to be valued around $51.14 billion by 2024, as we use in order to meet the growing consumer demand for clean level food and consumer packaged goods. So because of the awareness of the health and nutrition aspect of the food products, now that's why I told you this is the era of paradigm shift, where we're moving just to from traditional foods to nutritious foods. Whatever you eat, so we actually see what is the nutrition of that food matrix. So there are a lot of approaches to develop the clean level foods. So one of the approaches is your homogenization, high pressure homogenization. So homogenization term is very common to us. So when in the 1899, August Golin, when he developed the high pressure homogenizer for stabilization of the milk, so where he actually applied the high pressure to the fat components so it becomes stable for a long period of time without creaming off. So from that time onwards, so a lot of research on the high pressure homogenization is been done. So thanks to all the material science developments, like the homogenizer valve is very, very crucial or the heart of this high pressure homogenization. So how much pressure the pressure valve can withstand, that depends on the type of the material we select. So a lot of improvement, development has come on the material science area. So we could adopt that development in our design and fabrication of the high pressure homogenizer valves. So this might be known to everybody. What is homogenization? It's a process of converting the food particles to size good enough to provide benefits like product stability. The first criteria is to make the products stabilized. Stabilized means there is no separation of the phases in the food matrix. Uniformity, consistency, viscosity, shelf life, and improved flavor and color. So basically we refer the homogenization of any liquid foods means we refer it as a emulsion. Like there are two phases like immiscible phases. So when the two immiscible phases are combined, so sometimes because of their density differences, they move apart or they separate out from each other. So how to make them mix properly, make a emulsion? So that is our goal, and we can achieve that by homogenization. That means the breaking of the components or the dispersed components in such a way that it will be stable. That means the Brownian movement can easily stabilize these components. So it is accomplished by allowing the liquid foods to pass through a narrow valve gap by using high pressure. So when it is allowed to pass through a narrow gap, then what happens? We are constricting the passage of the liquid at high pressure. So when we are constricting the liquid or the valve, constricting the valve gap, then so there will be the increase in there will be maybe 100 times or 1,000 times of increase in the velocity of the liquid foods. So when the velocity of the liquid foods increases to 100 times or 1,000 times with the high pressure, so there will be shearing action. That means in micro level or nano levels also we can able to tear away the cells of the membranes of the food components. So as I have referred, that advancement means the advancement of how much of pressure we can utilize in the high pressure. Traditionally, or the conventional way of processing is to allow the food materials, particularly the liquids, at maximum pressure of around 50 megapascal, 20 to 50 megapascal. So advancement is referred as the how much of pressure we can build up or pressure we can apply. Like nowadays, even up to 400, 600 megapascal of pressure also we can apply. That is the advancement in the homogenization area. So enormous development in the design and manufacturing of homogenizers have been made to meet the demand. So what is the demand as per the homogenization is concerned? The high efficiency. What do we mean by high efficiency? The efficiency of homogenization means how much of the actual energy is being applied to the particular food component to do the work. So do the work means to break the particles, like from 3 to suppose 5 micron size to some 100 or 50 or 1 nano size material, right? So how much of the energy input is applied to the food components? That is our efficiency. But unfortunately, in the conventional process, a lot of the a lot of the heat is being, a lot of the energy input is being lost in the form of heat, and around 30 to 40% of the actual energy is being dissipated to the food material to do the job of particle reduction and other things. Right? So homogenizers performing multiple tasks is the latest development. So nowadays we don't see homogenization as only a way of stabilizing the food materials. Rather, we see a lot of other things in homogenization, like what other job it can do. Like, can it pasteurize the material also, pasteurize the milk also? So that is our question of thought. So not only it should stabilize the product, rather it should also pasteurize the it should be able to make the product safe, or it should make the product hygiene, right? So these are the recent trends of your homogenization. So as I mentioned, the development of the material science to withstand the very high pressure. Because of that, actually, a lot of development helped the design valves to suit the process conditions. So that I'll be discussing. So materials like the ceramic, the latest material they're using is the ceramic artificial diamond. So it's a comparison I am giving here to understand what is the difference between high pressure processing, the conventional homogenization, and high pressure homogenization. So because the high pressure processing is there in the research area, so quite some time. So people are working on high pressure processing. So with the scientific community, they are aware of that. So what is the benefit of the high pressure in the food materials? So now in the high pressure, we can achieve up to 600 or more than 600, 700, 800, 900 megapascal of high pressure we can give to the food material. So high pressure processing is a static process. It's a hydrostatic process where the food material is being pressed and in a chamber where the hydrostatic pressure is applied. Okay, so there is no shearing and no other thing because it is not, the food material is not moving in the process of high pressure processing. So since it is, the food is not moving, of course it will be a batch process. And the material handle, the earlier, the high pressure process, the systems, they were using very, they were able to handle very less amount of material. But nowadays, because of the advancement, so people can use the high pressure processing to produce around 300 or 400 or 500 liter of the liquid foods per hour. Not per hour, that is per batch. So that is the limitation of the high pressure processing. It's a static process, static pressure is applied, and it's a batch process. So since it is a batch process, so commercialization of the high pressure processing is, that is one of the hurdle of commercialization of the high pressure processing. Because cost of production will be much higher because it is a batch process. Now coming to conventional homogenization, so we apply a very mild pressure of 20 to 50 megapascal, and it's a continuous process. And because the food material is moving inside a small gap in the valve, so the shearing action takes place. So shearing action means the particles will squeeze or it gets elongated. So elongational stress, that we call it as elongational stress, being induced into it, and later part it breaks apart because of the the turbulence and the cavitation. Since we are applying very low pressure, so there is no preservation effect. So what do we mean by preservation effect here is whether we can make the product stable without the thermal treatment. So the answer is no in conventional process, because we are not able to kill the bacteria or the inactivate the harmful microorganisms into it with the use of conventional homogenization. So that is why the homogenizer is there and high pressure processing is there. Then people thought of why not combine these two. So in the homogenization, so they try to make it from batch process to a continuous process of high pressure processing. So people thought of that, and they have developed one new system that is called high pressure homogenization, where very high pressure is being applied in the upstream, on the upstream, and this high pressure fluid or the liquid will pass through a series of valve, maybe one valve, that is either it can be first one stage valve, one stage process, or the two stage process. So they combine both the high pressure and the shearing. So the mechanism is the high pressure is applied, and because of the high pressure is applied up to now the scientist could achieve up to 400 megapascal of pressure. And because of this high pressure, the shearing also will be very high. That means, so when the shearing will be very high, we can achieve a nanoscale materials in the food system. So when we can able to make the nanoscale materials, then so there will be a lot of benefit we can draw out of it. And because in the nanoscales also the particles can be ruptured, so even the bacteria and all these harmful microorganisms, they also get killed. So that's why the high pressure homogenization gives a preservation effect. That's why I mentioned. So we don't see the homogenization as a single process, rather it's a combination of two processes, like the stabilization and also preservation. So this is the advancement in the area of high pressure homogenization. So it's a comparison between the high pressure homogenization and the standard homogenization process. Like operating pressure, as I mentioned, up to 350-400 megapascal you can achieve in high pressure homogenization. In standard homogenization, up to 50 megapascal. The applications are like particle size reduction, the emulsification, mixing, food preservation. You see, the food preservation is the one which we target in high pressure homogenization, which we cannot achieve by standard homogenization process. So to achieve that, of course, the gap size should be very, very less, like 2 to 30 micron, or even less than that. And standard homogenization, it is 20 to 100 micron. So what are the commercial applications of the high pressure homogenization? It is still under development, with potential applications in dairy and pharmaceutical and beverage industries. So we could expect at least 10 years or 15 years or 20 years in future, we can see the high pressure homogenization will be using in the industrial scales. But the standard homogenization, so it has been a part of our processing operations, and mostly in the dairy. So this is the vital part of your pasteurization of milk and other products. The dairy, pharmaceutical, beverages. So a lot of use we can see in for this standard homogenization. So what about the microorganism and enzyme inactivation by high pressure homogenization? Yes, it is possible, function of temperature and pressure, of course. But effect of microorganism, effect of standard homogenization of microorganism is very, very less. Or I can say there is no effect on microorganisms. efficacy of microbial efficacy. Yes, we can achieve pasteurization even with little thermal treatment to the high pressure homogenization. So also we can, that means the synergistic effect of pressure and the temperature, we can even achieve the thermal sterilization, or commercial sterility also we can achieve. So what is the working principle of high pressure homogenization? So as you can see, the liquid food at a particular temperature, actually the process temperature depends that at what temperature we are feeding the material. So it's when it passes through intensifier, actually the high pressure pump is there, so it applies pressure to the pressure transmitting medium. So that actually this is an indirect process of achieving the high pressure. So 400, 500 megapascal we cannot achieve in the direct pumping system. So we use intermediate term or intermediate system that is called intensifier. The purpose of the intensifier is to intensify the pressure which is being given by the heat pump, and this intense pressure or the high pressure is given to the liquid food material. So the pressure is, the liquid is fed with very high pressure by the use of the intensifier, and that the pressurized liquid, that is T1, P1. So the pressurized liquid temperature is T1 and temperature T1, pressure is P1, is allowed to pass through a valve that is called high pressure valve. So the how much of the pressure or how much of the velocity we achieve, that depends on the valve gap. So this is where actually we can manipulate our process. So what should be the valve gap? What should be the design of the valves? So different valve design also give different shearing action or different cavitation action or turbulence. So it is allowed to pass through a very tiny gap with high pressure in the upstream. So because of that, so the shearing is happening. So it's a severe shearing action which takes place, and the velocity of this material at the valve gap will be manifold, like 250 meter per second or even more than that we can achieve with the pressure and the valve gap combination. So after that, there is a drastic change in the pressure in the downstream. So there is a drastic gradient of the pressure. So pressure gradient from P1 to P2. So if you are not using any secondary valve or the second valve, then the pressure P2 will be our ambient pressure. So there is like the difference, like suppose if you are maintaining 400 megapascal, so from 400 to 0.1 megapascal, that is atmospheric pressure. So there is a drastic change in the gradient in the pressure. At the same time, so there is drastic gradient in the velocity. And because of the pressure and the velocity gradient, so we could achieve, so we can achieve a lot of benefit in the food products. And immediately after this, so there is a cooling unit. So the product needs to be cooled. So where there will be temperature rise because of the shearing action, the input energy is given in the form of kinetic energy, and this kinetic energy is dissipated to the food materials, so to break the food components, and some part of that is utilized or is lost in the form of heat. That means there is an increase in the temperature. So it's a thermodynamic relationship like pressure volume. So immediately you need to cool it down so that the after effect of the high pressure homogenization should not be there in the food products. So if you are using the low pressure valve, actually the low pressure valve, the purpose of the using of low pressure valve is to sometimes what happens after homogenization, so there will be clustering of the particles. On the clustering, the particles are there. Again, there will be chances of coalescence, and they may get separated out from this food system. So we require a little back pressure, or you require one secondary valve to break those particles. So after that, we again cool it, and we can process it aseptically also, because since we are achieving the pressurization effect or the sterilization effect, so we can store it directly to the aseptic packaging materials. So a little insight into what is the mechanism of the action in the high pressure homogenization. So in the valve gap, what happens? There will be shearing action, as I mentioned. So the shearing action, why the shearing action happens? Because there is a drastic pressure gradient from 400 to 0.1 megapascal, and because of this drastic pressure in gradient, that means the pressure force or the pressure energy is converted to the kinetic energy, and because of this kinetic energy, so there will be the shearing effect. So the shearing effect is characterized by a term that is called Weber number. This is a ratio of how much of energy we are inputting, that is the inertial force, to the surface tension force. Then the shearing of the particle also depends on the surface tension of the inherent characteristic of the food material. It should actually surpass the surface tension force to break the material. So the Weber number decides whether, or a critical Weber number should be there, where beyond this number, your fat particles or the particles of our concern gets ruptured. So this is the way actually we characterize this Weber number. So at what Weber number we can able to break the particles. So this is actually, traditionally it is in the standard homogenization, we actually do that. So Weber number, the shearing effect, which is represented by Weber number, depends on the surface tension force, the density of the particle, and the velocity, of course, and the length of the valve gap. So these parameters, like the density is okay. So, but so in standard homogenization, all these processes all the properties actually we can measure, and accurately we can calculate the Weber number. But problem with this high pressure homogenization is because all these properties are also dependent on the pressure. That means with the high pressure, these properties changes, like the surface tension. The surface tension changes with the pressure. Also, the density also changes with the pressure. So since they are dependent, or the properties are dependent on the high pressure, then this conventional way of defining the shear effect will not be possible. So that's why. So now our research goal is to how best we can determine all these properties accurately, and so that we can exactly say that the shearing effect is the only effect, or is the sole effect which actually ruptures the particles. Yeah. So then coming to cavitation, what cavitation is, actually there is a pressure gradient, so drastic pressure gradient, and pressure will be so low enough, sometimes it comes to the level where the vapor pressure of the liquid passes the atmospheric pressure, or it surpasses the atmospheric pressure. That means there is a phase transition which is happening inside the valve gap, and because of this phase transition, so there will be cavity formation. And this cavity at the later part of the valve gap, it collapses. When it collapses, then there is a shock wave that gives to the adjoining particles. So the shock wave in the high pressure homogeneity is so high that it can also not only it can rupture these the particles, but it can also activate some of the components, which also like some of the components it can be activated, which can also give the ill effect to the food materials. So this is the cavitation where the cavitation depends on the pressure gradient, and of course the pressure gradient we can manipulate with the back pressure. So what amount of back pressure is good enough to initiate the cavitation. So there are two terms, either the cavitation number or Thoma number, that is P2 by P1. P2 is the downstream process and P1 is the upstream process. But inception of cavitation is very, very important. So at what point actually our cavitation is occurring? At the upstream, or at the middle of the valve gap, or at the downstream? So it is being seen that since actually this is not only dependent on the pressure, also it depends on the velocity. So that means the half rho v square, that is the kinetic energy. So it's a ratio of the pressure energy to kinetic energy. So inception number, the cavitation inception number will tell us at what point actually our cavitation is happening. So whether it is happening at the downstream or upstream, or at the middle of the valve gap. So cavitation actually occurs at the downstream, just at the exit of the valve gap. So this is what we have, the scientists have proved. And the extent of cavitation, the how much of cavitation force will be there, that will depend on the cavitation size. And what is the size of particle which will be broken, that will be dependent on the cavitation size, or the cavity or the bubble size. The same way your turbulence is also one phenomena where, because of the high velocity, like 250 meter per second, or even we can achieve the supersonic velocity at the small valve gap. So the flow profile will be completely changed. So from laminar to turbulent, and because of the turbulence, the dissipation of the energy also will be good. So, but again, this turbulence depends on the product property, like the surface tension and the density. And also, so there is another term, that is epsilon, that is your energy supplied per unit time. That is like energy density and the rate of energy dissipation. So all these parameters are related, and all these parameters decide what is the maximum size of the particle which cannot be, what is the minimum size of the particle that cannot be broken up. So this parameters determination is very, very difficult, because we have a very small valve gap, and the residence time is, you can say, microsecond or millisecond in the valve gap. So with this short span of exposure to at high velocity and high pressure, so it is really difficult, the top task for the scientists to actually characterize all these phenomena. So people have adopted the numerical simulations process of defining the velocity profile. So, but again, here one problem occurs is the lot of assumptions actually people make while they do numerical simulations. So a lot of advancement has to be done, or a lot of research need to be focused on how realistic data we can take to model our system. Like we define the velocity profile and all this, so that our design of the valve will be good, or will be efficient one. So there are different valve types. the counterjet valves. So the counterjet valves is one where the upstream liquid is divided into two small channels at very high pressure, and these two small channels, the liquid in the small channel again, they meet at the downstream with very high pressure. So there is one term called impingement, the process of breaking of the particles in the counterjet valves is the impingement. So mostly the microfluidizers, which are generally used for the pharmaceutical use in the laboratory, so they work under this principle. That means how much pressure will be applied to the system will depend on the velocity of these two streams. Then very small channels are there. So smaller the channel, more will be the viscosity and so on. So this is called counterjet valve. So it is a static system. There is only the liquid is moving into two separate channels, and again it meets, and due to the impingement, the particles breaks down. So another one is radial diffuser valve. So where the particle which moves to the valve are moving radially. So there is a valve gap and valve seat. So the upper one, that is the valve seat. So the valve seat, the gap of the valve seat will decide how much of pressure it can generate and how much of velocity we can achieve and how much of shearing and other phenomena can occur. So that will be decided based on the gap of the valve and the valve seat. So another one is the axial flow through valve. So the materials actually pass through the valve axially. So in the axial direction, actually it passes. So through a small channel, it passes around like 20 or 0.20 microseconds or that kind of time. So when it passes through that channel, then it meets a pin valve. The pin and pin and the pin valve and seat. This is called pin valve and seat. So this is one kind of actually valve. So it passes through a narrow gap, and this narrow gap is controlled by the position of the pin. Okay, so there is an actuator actually. So where the gap of this pin will be controlled. So this is axial flow through valve. So there are a lot of these are actually basic three categories, and these are some of the valves, different types of valves, and they have different use also, because the valve is also one parameter where it decides the extent of your or severity of the pressure treatment. So some of the common valves are like the nozzle, conical piston, needle seat, microfluidics, the ball and seat valves, sharp angles. So they have different purposes. Like some only do droplet disruption, microvalve inactivation, cell rupture. Even some can produce in micro scale, some can produce in nanoscale materials. So all these differences are there. That means while we design, so the purpose of giving this information is to first see what is the type of the valve we are using in the high pressure homogenizer to achieve the target goal. So what is our purpose? Suppose our purpose, whether it is to denature the protein. So if it is denaturing the protein, all the valves cannot be used for denaturation of the protein or the breakdown of the protein structures. So that selection actually we need to take before designing our system. So these are the common characteristics of the high pressure homogenization valves. Like counterjet, as you can see, in the counterjet you cannot use more than up to 200 megapascal. That means in microfluidics, so the limiting pressure till now is 200 megapascal. So flow range controls the operating pressure. So non-movable part in this. So it is, of course, a lab scale process, as I mentioned. So microfluidics or microfluidizers are used only for the pharmaceutical use. In most of the pharmaceutical life cycle, we can see microfluidizers. Then coming to radial diffusion valves, different companies, they have a lot of companies are now in the market. They produce the high pressure homogenizers. So they can achieve 150 megapascal of pressure. So both life scale and industrial scale production also they can. So people have done. So here in this case, flow rate depends on the operating pressure. The flow rate depends on the operating pressure, how much of pressure we give. That means through the intensifier. So that will decide the flow rate. And in this case, because the shearing is more severe than the counterjet, the temperature rise is about 17 degrees Celsius per 100 megapascal per watt. And axial flow through orifice, so we can achieve up to 400 megapascal of pressure. So lab scale and the pilot scale systems are available, but till now there is no industrial scale of high pressure homogenizer using axial flow through orifice valve. The flow rate in this case is constant. Its pressure is controlled by adjusting the cap size, and the temperature rise as it will be more severe than the radial and the counterjet. So the temperature rise will be 20 degrees Celsius per 100 megapascal of pressure applied. So this rise of temperature in the earlier slide actually also depends on the type of the material we use. So whether it is a viscous material or a liquid-like material or the water-like material. But most of the cases, while designing our system, what we do is we assume it as a Newtonian fluid. But it is not always the case. Actually, as you know, most of the poor metals are not Newtonian, non-Newtonian. So it is very difficult to characterize these terms or the. Actually, we do a lot of assumptions in calculating all this. Or we designing the system. Or designing the process. So, as we are talking about high pressure homogenization, and one of the purpose of the high pressure homogenization is not to stabilize the system, rather also regeneration effect also should be there. So temperature rise or the relationship between temperature and pressure has to be properly, thoroughly studied, and how best we can maintain the temperature so that we can achieve the bacterial death, or we can achieve this even inactivating the spores. So that will depend on the how much of temperature we maintain during the process. So there are two terms. One is heat of compression. So heat of compression, that is because of the compaction of the materials or the components. The heat of compression rises. That means because of the pressure only, your heat of or the temperature rises around 2 to 3 degrees Celsius per 100 megapascal. So it's a reversible process. When we depressurize the system, again the temperature will go down. So it's a reversible process of temperature rise. So around 2 to 3 degrees Celsius rise because of the heat of compression. But of course, that also depends on the material, as I mentioned. So every material will not have the same heat of compression. Then heat of homogenization, because of the very high pressure or the pressure energy is converted to the kinetic energy. So this kinetic energy is dissipated in the form of the heat energy, and that heat energy is at the range of 14 to 25 degrees Celsius rise per 100 megapascal. Okay, so whether this is the area where we need to decide whether we are targeting the or whether we are calling the system as a non-thermal process or the thermal process. If suppose we maintain the temperature above 100 degrees Celsius, then people will be skeptical to define it as a non-thermal process. But there are a lot of actually argument goes on. Even though we maintain more than 100 degrees Celsius, but the exposure time is very, very small, less, like some in microsecond in microsecond actually we expose the product to at very high pressure or very high temperature. So still people they call it as non-thermal process. But generally what our actually perception is, if the temperature is above 60 degrees Celsius or 50 degrees Celsius, we don't call it as a non-thermal process. So this heat of homogenization is dependent on many parameters, like the final temperature, depending on, like, there are a lot of empirical equations actually taking place. Empirical equations actually people have developed. So the product temperature, how much product temperature will be achieved, will be dependent on the inlet temperature, and the plus of del is the heat of compression, and the zeta is the heat of homogenization, and some amount of heat loss. So to make the system more efficient, the heat loss should be minimized in the system. So we need to give it the cooling wrapping or the cooling pad in the valve we use, so that the heat loss to the environment will be less, and so all the energy which will be input will be given to the components which will be broken down. So as you can see, there are different stages, like from tank to the initial heat exchanger. So the temperature is rose to 75 degrees Celsius. Then this is the effect of heat of compression. You see, so heat of compression, even though the heat of compression per 100 megapascal, it is increasing, but here it is decreasing, because actually, so heat loss is taking place. The equilibrium has not reached at this point. So in the compression stage. So when it passes through the first stage, then the temperature rises. And the holding time also very, very crucial. So then I'll be discussing what is the importance of the holding time when you actually target the sterilization effect. Then holding time is very, very crucial. Then immediately after that, so product is to be cooled down. So in the second, the bottom one, in the graph, you can see that the heat of compression, the dash line is the heat of homogenization, and the solid line is heat of compression. So this is the difference. So very huge difference, like the initial product temperature was 90 degrees Celsius in this particular system. Now they could achieve up to 150. That means there is a 50-60 degree rise in the temperature. So what is the purpose of giving this is that we need to design our system in such a way that our product should be minimum, or we should actually achieve a predefined temperature. Like suppose we are targeting the spore forming, the killing of the spore formers. So nowadays with the high pressure homogenization, so people use temperature up to 140, 150 degrees Celsius. But the recent advancement is that, so even at 110 or 105 degrees Celsius rise, we could achieve the killing of the spore formers. So, but also it depends on the types of the spores and all these things. So initial product temperature, pressure, valve design selection is crucial for attaining the final process temperature. This is where our research should be focused on, that what is the target temperature and how we can maintain it. And that is very, very crucial. So what is the difference in the temperature increase with the processing? Like high pressure, heat of compression during high pressure processing, and heat generation during the high pressure homogenization. So of course, high pressure, heat of compression by hydrostatic pressure, heat of generation, heat of homogenization due to the discharge through the valve. The principle is reduction in the intermolecular distance. When the intermolecular distance comes down, then your heat of compression will increase. So here the kinetic energy converted to dissipation energy. So dissipation of the kinetic energy. Also the dissipation also depends on the mechanism of action, like how much of cavitation, how much of turbulence is there. More turbulence is there means more energy dissipation into the system. So what are the factors like heat of compression? The factors like the composition, as I mentioned. The composition also decide how much of heat of compression will be there, the pressure and the product initial temperature. Heat of generation is irreversible and it depends on the pressure and the valve design. The flow pattern is one area where we have not yet been so successful, I can say. So, we have not been so successful because, as I mentioned, because we're handling a very, very small, we're targeting the area where a very small gap will be there and the velocity will be very, very high. With a very short span, like in microsecond, actually all this phenomena takes place. So there, characterizing the flow pattern is very, very difficult. So only way to characterize this flow pattern is, that means experimentally we cannot determine all these flow patterns, like what will be the flow at a particular area at the beginning or at the entrance of the valve and at the exit of the valve. It's very tough. But scientists have used the computational models, computational fluid dynamics. They have used and they use it extensively to characterize the flow. And the models are based on the many assumptions which fail in realistic condition. That's what I wanted to say. That because in the realistic model, in the lab scale, somehow that will be good. But when you go up for scaling, then there will be problem. So what is the problem? What is the advantage of knowing these flow patterns or the characterizing the flow patterns? This is actually we can infer, actually we can say what is the recirculation, what is the zone of cavitation, all these things, so that it will help in designing our valve. Now coming to the high pressure homogenization induced changes in the food structure, I'll cover the overview of the things. Actually, I'll not go in detail to individual food products and how that is happening. So, about a century or above little more of century years ago, the Nobel laureate Dr. William Bridgman actually. So in 1914, so he did an experiment with the high pressure, and he did an experiment with the egg white. So he pressurized the egg white. So when he pressurized the egg white, what he could achieve is that the egg white, the density or the consistency of the egg white increased, and it was like a gel-like structure he could get. So for his contribution to the insight into the protein structure and all those things, actually he got the Nobel Prize in 1945 or somewhere around there. So from that time onwards, actually, people, particularly in the proteomics area, people are concentrating on the changes in the protein structure with the high pressure. So this is the very basic things actually I have given. As you know, there are 4 structures like the 4 structures like primary, secondary, tertiary, and quaternary structures. So what do you mean by the denaturation is to unfolding of these structures. And as you can see, different bonds are there. Like in the primary structures, we have some peptide bonds of amino acids, which will give you a peptide chain. So in the secondary structure, we can have some hydrogen bonds between these peptide chains, so that it can give the helix, alpha helix structure or beta sheet structure. Then these secondary structures are combined with each other by disulfide bond to give a tertiary structure. And all these, so more than 2 tertiary structures, they combine, they rearrange themselves to give a 3D structure that is called quaternary structure, which is very common in our food products. So at what structure, whether it is a primary structure or secondary or tertiary or quaternary structure, that will decide the functionality of the protein. So there are a lot of functionality of the protein, like solubility, swelling, water holding capacity, like gelation also, foaming, emulsifying, and flavor binding. So disruption and the reformation of hydrogen bonds, rupture of hydrophobic interactions, separation of ionic pairs. So the ionic pairs also we call it salt bridge. So all this depends on the structure, the pressure, temperature, pH, ionic strength, solvent composition, protein concentration. So, and here on elliptical phase diagram I have given. So the dome kind of structure, actually the change of the protein structure dependent both on pressure and temperature, what we can see here, and also the volume change. When the volume change will be there, so there will be the denaturation of the proteins. So there are a lot of theory of the denaturation, and even people have done a lot of work on how much of energy input should be there to convert a quaternary structure to tertiary structure and so on. We can see here protein denaturation is a function of pressure. In conventional denaturation process, in the bottom one, there is an abrupt change in the structure, like with the temperature, with the use of temperature from quaternary structure to like secondary structure and all. So the change is abrupt. But in high pressure homogenization or the high pressure homogenization process, the state of the protein will gradually unfold, like native to early folding or intermediate stage, then unfolded state. So people say that, so since it is the change is gradual, so we can easily manipulate the protein functionality better compared to the thermal process. And as far as the homogenization is concerned, so we can achieve or we can manipulate or we can change the quaternary structure at above 150 or at around 150 or less than that pressure. Tertiary structure, we can destabilize or we can unfold it at more than 200 megapascal, and secondary structure is above 300 megapascal. But still, secondary structure is the unfolding of the secondary structure is very, very tough, though. Actually, that will decide so how much of effectiveness of our homogenization pressure is on the like different types of enzymes, like identification of the enzymes. So what is the effect? So here it is showing some at different pressures how the proteins are behaving, like shear induced increase in temperature. So at 0 megapascal, the protein structures are there in the colloid forms. You see the particles size decreases at 200 megapascal, but it's peculiar things that is being seen at high pressure is the combining or the clump formation. And this is due to the shear-induced increase in the temperature, which favor the hydrophobic interactions among the individual casein proteins. So because of the hydrophobic interactions, again they combine, leading to mycelius reformation at homogenization pressure of 250 megapascal. So there will be hydrophobic interactions happening at high pressure. So again, a lot of in-depth research is required. So exactly at what pressure that is happening and what are the parameters which influence these things, like even valve design also will affect these hydrophobic interactions. So that is our thing. So as you can see, there are some references, like so people could achieve the gel strength of the milk by use of the high pressure homogenization and increase product yield in cheese. So actually what happens in the absorptivity of the protein particles onto the fat surface or fat membrane surface is much higher. So when the fat particles or the protein particles are adsorbed to the fat particles, then the loss of the protein to the whey will be less. In that sense, they can say that yes, there is an increase in the cheese production. Like they can achieve up to 12 or 14, 16% of protein yield. Like 17%, 12%, 32% yield with different pressure. The particle size reduction, generally we see when we talk about the milk component, the milk fat. So, if you take the graph of the particle size of the milk, so we get it two dimodal or bimodal structure actually get. So one is for the fat particles, another one is casein mycelius. So there are two bimodal structures we can see. So when we apply pressure, then what happens? The bimodal structure is converted to a single mode, but the shift is initially actually there is the particle size distribution or the span will be very, very large. Like as you can see, the black line, solid line, the fat globule size in the range of the 1 to 10 micron. But when the pressure is applied, there will be shift of the modal structure. So first the bimodal structure will convert to single mode, and single peak is there. So now from 1 to 10, the range it comes down to less than like 0.01 to 0.01 to 1. So there is a shift in the particle size distribution, and the narrow range we can achieve. So when the consistency and all this uniformity we're talking about, that depends on the particle size distribution. That at this particular particle size, the distribution will be proper or uniform, or we can say like that. So unfortunately, 35 to 59% of the input energy used for particle disruption, but you see 63 to 41% of the input energy lost as heat. So this is where actually our design intervention has to be there, that how much of heat has to be, how much of energy actually should be given to the particle to get the job done. So it's not be lost in terms of heat. So as you can see in the right hand side, there is a graph that is the pressure of 300 megapascal. So a single peak is there, but there is a tail. So there is a tail. So that is that depicts the clumping of the components, the fat components or the protein components. So because of the hydrodynamic interaction, that's why that tail portion is coming. the HPH treatment effect on the emulsification, as we know, so that the coalescence we don't require. So our products will be stable. So this stability depends on two things. One is van der Waals forces of attraction and electrostatic forces of repulsion. So here also we can see in the graphs, the micrographs actually. So at 20 megapascal, the size are bigger, and 200 megapascal, so we could achieve in nanoscale materials. But above 200, again, there will be some clumping. That's what it is being depicted in the graph. So there is hydrodynamic interactions. So even all these inferences are actually some guessing or some guessing kind of thing. Like people actually they have not yet understood the actual mechanism of this clumping. So this is where actually we're in. So now our focus has to be there at this point, that why that is happening at a very high pressure. So it depends on different energy, different parameters like the energy density, which is a function of homogenization pressure and the flow rate, flow conditions like laminar or transient or turbulent, valve design, properties of the emulsifying agents, including and the types of concentration of emulsifiers, stabilizers. The microbial inactivation, as you can see here, at standard homogenization, it is not possible to achieve the microbial inactivation. But see the right side at above 200 to 600 like that. So we can achieve the microbial inactivation, like energy enzyme inactivation also we can achieve microbial destruction. So we could achieve pasteurization and sterilization condition also, nanofragmentation, enzyme interaction, enzyme inactivation, nano covering also, nano encapsulation also we can do with the high pressure homogenization. There are different factors to inactivate the microorganisms, like the machine factors like pressure intensity, pressure temperature, process temperature, residence time distribution. People have not yet given more emphasis on the residence time distribution, but this is very, very important. Number of passes, the product is like pH composition, viscosity. Viscosity has not direct relations with the microbial inactivation, but viscosity will decide the residence time of that liquid in the valve gap, and that will decide the final lethality of the process. The vegetatives, or whether it is vegetative or spore formers, as you know, the spore formers are more heat resistive. Gram negative are more prone to heat and pressure effect. Then even some the effects are strain specific. Some strains may not be properly effective to high pressure, or high pressure may not be effective to a particular strain. But with the same species, other strain will give a good result. So it has been found out that inactivation of the vegetative organism can be achieved with modest condition of like less than 200 megapascal with less than 50 degrees Celsius. That means non-thermally we can achieve the killing of the vegetative organism. But nowadays, more focus is on the inactivation of the spore-forming bacteria. So the spore-forming bacteria, the intensive pressure thermal condition actually they give. So generally they try to maintain pressure above 200 megapascal, and the temperature of process, that is TP, should be above 100 degrees Celsius. But there are still some lacuna in that research, that what is the thermal, what is the chemical marker for all these processes, like hydroxymeryl HMF production. So that chemical markers are still actually lacking in high pressure homogenization process. So that we can say that yes, you see the, like our traditional way of saying like pasteurization is 72 degrees Celsius for 15 seconds, but we cannot define pasteurization in case of high pressure homogenization. Okay, so there are still a lot of things to do. As I mentioned earlier, the fluid flow, it depends on the fluid flow characteristics, HPH residence time, flow rate, heat transfer, and heat transfer also dependent on the flow conditions, like the turbulent flow or cavitation is there, or which is predominating. So there are some of the research. So enormous research has been done on this killing of the bacteria. Like Geobacillus stereothermophilus is considered one of the most pressure resistive bacteria. So 2 to 5 log cycle reduction with this particular condition, like as you can see, pressure above 350 or 300. What type of valve we can use? Like yeast and mold, we can use a little milder process temperature, the process pressure, like 130, 200. Salmonella, as you can see, so 3 log cycle with buffer, with different buffers also, they have found out different things, different results, and different types of the valve they used. So I'll give a complete overview of what are things happening or we can manipulate with high pressure homogenization, which cannot be achieved by high pressure processing. Like operating mode, as you know, continuous pumping, treatment time is 0.2 seconds, even less than that. The treatment time in this high pressure processing is 3 to 10. The effect on covalent bonds in small molecules is sensory impact. In high pressure homogenization is ineffective. Also in high hydrostatic pressure, it is also ineffective, because the covalent bond, if it is broken, that means we are not able to break the secondary or the primary structure of the protein, or the modes and yields highly effective at this pressure. Bacteria highly effective compared to hydrostatic pressure. Oxidative enzymes strong in inactivation at more than 300 megapascal. Antioxidant capacity increased to 150, more than 150%. That means, so what it means that we need not provide any additives to give the antioxidant activity or antimicrobial activity to the food materials. In that way, actually, we can say that the product produced by high pressure homogenization is clean level food. Thermal marker, as I have mentioned, is not yet detected or not yet researched on. The vitamins, coming to the nutritional part, the vitamins, most of the vitamins are preserved. Like aroma compounds also there and unaffected. That means more real-like or the fresh-like feeling actually you can get with the high pressure homogenized processed juices. All this actually, most of the cases actually see the high pressure homogenization, the property is increased and there is less degradation of the nutritional components. So in a nutshell, what are the opportunities and challenges? Like more insight into the fundamental aspects like the forces of cavitation, shear and turbulence is required to better understand how these phenomena affect the product quality. The more fundamental research is required. As I mentioned, in situ determination of the flow parameters is quite challenging because we have a very small residence time. So computational models may not work in industrial scale HPH system that you need to look into, because till now there is no industrial scale production, but when it happens, then we need to validate the computational models which exist in the lab scale, whether they work in the industrial model or not. Scale-up process needs validation of parameters, the researchers claim. The fundamentals on spore inhibition by HPH is still lacking. So what is the fundamental of the spore inhibition? Still it is lacking. Pressure thermal uniformity during the HPH is one of the areas which we need to look into. Identification of the process indicators will design the process giving better product quality. Of course, another one challenge is the cost of production. But so as I mentioned that with mass production, the cost will go down. So with the hope that the HPH will be very popular and the cost of production will go down when the mass production will be there. So I'll conclude my lecture with this, that high pressure homogenization is a promising food processing operation that can be scaled up for industrial continuous operations. It is validated that HPH can preserve the food quality better with mild heat treatment. Structural modifications of food components gives better functionality. Foods can be preserved with minimum or no use of chemicals, additives, so as to qualify for clean level tag. So thank you very much. So this is the pictures where I am working, my university, Ohio State University. So thank you very much for giving me this opportunity to share my little knowledge with you people.
Thank you, sir. Thank you so much.
Yeah, any questions, please?
Very good, Dibyakanta.
Thank you, sir. Thank you.
I think it is a very good presentation. People should have that. I think किसी को कुछ question होगा तो Anand Paul. If there are 2 questions, Jayashree, you can read or they can themselves ask.
Yes. You can ask your question directly, Mr.
Okay. The question I have is, if protein structure can be changed, so why can't we make imitation dairy products with high pressure processing? So if there is a constraint, what is the constraint you foresee, sir, on imitation dairy products to be made via high pressure processing techniques?
Yeah, that's why I told you that can be possible. I cannot say no, that can be possible. But a lot of research need to be done on that. So we can imitate the dairy products also.
Thank you, sir. Thank you.
Yeah.
Thank you, sir. Is audience ready for other questions? I think the lecture was so elaborative that they are very good questions.
Maybe I
do you have any questions?
I think there was some question from Shubham.
Mr. Shubham has completed. Shubham?
Thank you, Subham. But a lot of things are there actually, which actually this is beyond my actually area. Like a lot of in the microbiological aspect, so many things are there. Even the chemical aspect, biochemical aspect, so many things are there actually. So a lot of review papers are there. So if you are interested, then you can go to the individual review papers. So they have a very elaborative discussion on these things. And one thing I need to mention here that there are 2 in the world actually as far as the killing of microorganism or the spore former is concerned. So another one focus area in US is first they try to in the Europe actually what they do, they first try to germinate the spore formers and they give little mild treatment to kill the spore formers. But in US condition, so they target differently. They don't germinate it because people claim that even you do some processing, you cannot germinate 100% of the spore. Yes, it requires a high process, or the synergistic effect of heat and pressure has to be properly studied on.
I think we can proceed further. Coming to the end of our 25th webinar by Dr. Dibyakanta Seth, I invite our research scholar N Mishra to give vote of thanks to Professor Seth. Saurav?
Thank you, Deep. A very good evening to all. It gives me immense pleasure to propose vote of thanks for the 25th session of FCTL webinar series. I wholeheartedly thank our speaker, Dr. Dibyakanta Seth, for the wonderful session and delivering brief insights on various aspects of high-pressure homogenization from the preservation point of food products, and such as particularly level food and the difference between high-pressure process and high-pressure homogenization. What is the working principle, as well as at the last he emphasized on the effect of HPH on different qualities of food product as well as microbial inactivation process. So thank you very much, sir, for giving us valuable time and guidance. I express my heartfelt gratitude towards our mentor and coordinator, Professor N Mishra, sir, for planning and arranging this webinar session. Thank you so much, sir. I would like to thank all the FCTL family members, participants for attending the webinar session and making this event a grand success. Thank you so much, everyone.
Thank you.
Thank you, Saurav.
Bye-bye.
Our next webinar, the 26th webinar, is by Dr. Gayatri Mishra on 23rd October. We will release the topic and the times shortly. Thank you all for joining. Have a joyous and festive